Anybody have any questions while I'm getting this set up about anything? Okay, we have, in case you don't know, for some reason, we have lab today. You do have to have your lab coat and you do have to have closed-toed shoes. If for some reason you didn't have those today, today is kind of, you get a pass. But from here on out, if you don't have a lab coat, I do have a few I can loan people if you forget them now and then. But you can't rely on me providing it to you every day. And with closed-toed shoes, like we can assess the situation if you forgot to wear appropriate shoes. Like if they're flip-flops, you cannot come to lab. So, but something else we can see if, you know, we're just, it's fine to take the risk. It's up to you and whatever. But yeah, what are you talking about? What? On top hat, on the actual like pre-labs?
It just says exercise line, aseptic techniques, and like I'm reading through and there's questions, like there's a question one.
Yes.
Answer it, and that already sends it to Canvas.
No, so this is the thing that some people get a little bit confused about. So on the assignment on Canvas, it'll even say like you have to complete this in Top Hat and your grade will not transfer to Canvas. I have to do it by hand, like each grade by hand. So those will absolutely not be posted on a regular basis. I try to get to them once a week, just so you guys know. But if whatever it says in Top Hat typically is what it's going to say when it gets over. Now, this first exercise, I always forget to edit it. It's like one of those things that like I start seeing the grades post. I'm like, oh crap, I forgot to change this because there's only 5 points for this assignment on Canvas. And there are actually 10 points available on Top Hat. So typically what I do, as long as you have tried to answer questions in both parts, if you don't know what I'm talking about, then you probably don't have questions answered in both parts. So there's 5 questions in like one area, and there's another 5 questions in another area. If you've only answered 5 questions, you've only got one part taken care of. So basically, if you have effort put into one part, then you get half the credit. And if you have effort put into the other part, you'll get half the credit. And so it's either you're going to get a 0, a 2.5, or a 5, basically, is what I'm saying. So I'm going to give full credit if you put effort into a half and effort into another half. That makes sense because of how I messed up the grading there. So pretty much if you try, you're going to be okay on that one. So think of it as like a primer. Get you oriented. Yeah. All right. Zoom. Okay, sorry this is taking forever. I forget how long it takes every time. Is this okay? Oh, I already have this going. Okay, I was like so confused. I was like, why can't I start the Zoom? Y'all. Okay. Okay, we're gonna do the Kahoot first. You guys know kind of how this is gonna go now. Jeez, man, there's so much I have to turn on and stuff before I can get started. So chapter 2 review or whatever, you know, Kahoot review. Let's get going. And then we'll talk about chapter 4. All right. Okay. All right. All right, I'm gonna go ahead and get us going. If you guys ever, so if you're ever joining in on Zoom, you can always join in here. This is one of those things that you can participate with everybody who's here. in class. Also, if you come in late for any reason, you can always join that number, 8580952. That'll be at the bottom for most of the screen, so you can always just join in that way if you want to. So we're gonna go ahead and get started. All right, which type of chemical bonds involve sharing electrons? We're going to knock this light off up here. So exactly, covalent bonds, that's the sharing. And if the way I remember is it says co. So I feel like that's like cooperating, couple, whatever. So working together. All right, hydrogen bonds. What about these guys? What is this paper? All right. So hydrogen bonds. Hydrogen bonds are the bonds that are going to hold the water molecules to one another based on the polarity, right? So oxygen is hogging the electrons, it's hogging the Barbie. So it has that negative polarity to it. And that makes the hydrogen that it's attached to have positive polarity. That positive of the hydrogen, if it associates just by attraction to anything else that has a negative polarity or charge, that is a hydrogen bond. So there's no actual exchange of electrons. And then like the popular choice people chose here, forms between hydrogen atoms in different molecules. It's not hydrogens that are interacting with one another. That would actually, if we're looking at this, like if the situation tends to be, and I say tends, but is always going to be the case that when we're talking about a hydrogen bond, the hydrogen has a positive charge. So positive isn't going to be attracted to positive. So it's going to be attracted to negative. So that's how we know it's not going to be between hydrogens. All right, what element is in all organic molecules? The main element. All right, awesome. So carbon is an element that's going to be found in all organic molecules. But remember, you're not technically organic unless you also have hydrogens associating with it. But the carbon is a big take-home message. All right, true or false? Water is a poor solvent. All right, so it's false. Water is the most important solvent of life because of that polarity we just revisited. What is water's pH? The pH of water. It's going to be 7. That's our neutral for all of this. Remember, our extreme at the acid end is 0, and our extreme at the base or alkaline end is 14. And it's all going to have to do with the ratio of the hydrogen ions compared to what should be the equal amount of hydroxyl or OH negatives. Should be equal in just plain water. All right, what range on the pH scale is an acid? I guess kind of give it away. That's all right. Alrighty, so that is going to be a 0 to 6.9, anything less than 7. All right, lipids. Let's see what we know about lipids. All right, so yep, this is going to be all the above. Great job. We already know they're going to make up the cell membrane. We talk about phospholipids. I mean, it's in the name. We also have used to make hormones. So a lot of the hormones are going to be based on sterols, like cholesterol and stuff like that. So those are lipids, and can be used to make energy for the cell. We know that we store fat to store energy. So that is what that's referring to there. We can break down fat and get energy back out of it. All right, which of the following organic molecules is amphipathic? Okay, so it's a phospholipid. Remember, that's going to be the phosphate group with that negative charge, and the lipid is going to be hydrophobic. It's going to be nonpolar, water-fearing. Got both going on. That's what helps it build like the main structure of lipid bilayers for our membranes. All right, what is a monomer of a nucleic acid? All right, so we're talking about nucleotides here. A nucleotide, remember, that's going to be a phosphate group, a pentose sugar of some kind. It could be deoxyribose or ribose, depending on what we're talking about. And then a nitrogenous base. That's what makes it A, T, C, or G, for example. All right, last question. Alpha helixes, or helices is what it should say, beta pleated sheets are examples of which type of protein structure, which level of protein structure? Alrighty, so this is one that we just got to take a little bit of time to remember which one is which, but primary is just going to be the amino acid sequence. That's just the ribbon that we made. Secondary is going to be literally the amino acids right next to each other, interacting with each other. And that creates the alpha helices and the beta sheets. Then we have the tertiary structure where those larger structures can now cause interactions like across the way, not to the guys just next door, but guys like, across the ribbon, basically, creating that initial folding overall of that specific ribbon. But then we can have groups of ribbons coming together to make a functional protein or enzyme or whatever it is. That's the quaternary structure. Right, let's look at our podium. Let you guys bask in the glory of it for a little bit, and then we'll get started with the lecture. Let's see if I can get this to work. Good job. All right, close this out. Just this one. There we go. All right. Oh, I didn't hit the dual push in time. You gotta be freaking kidding me. It's gonna be the death of me. Well, the death of this computer. Doesn't really matter if I click if it's my device or not. It's not gonna remember anyways. So, oh, it wants my password. Let's go to once. Just didn't know the right thing. That's awesome. Promise I'm gonna figure this out one day. Okay, here we go. Play. Okay. Zoom. Let's switch. want to just do this one. I don't know how. This is the frustrating thing is I got this to work and it was fine. It was good, but this is actually different than my computer. So now, of course, it's not going to work. So that's awesome. Yeah. Switch. This up on the screen. I don't want to switch to do this. Yeah, this is not the same as what I did or whatever. I guess I'll just have to do it on this screen and not on my phone or my tablet like I wanted to, so I could actually like write stuff for you guys where everybody could see it and have it. But like I this is completely different from my computer's way more updated than this one is. So I don't know. It's only letting me switch between, it won't let me like co-host or whatever. It won't even let me look at it. So whatever. So I'm trying to think. Okay, well, whatever. We'll just do it the normal way. And then next time, we'll see if I can figure it out. Anyways, chapter 4 is dealing with bacteria. We're going to learn about the parts of the bacteria and how they function. And this is why we have to give that sort of chemistry overview, as well as like the rundown of basically what a cell is and all of that. So it'd be nice if I could draw this on the screen, but this piece of junk isn't going to allow that. So I'm just going to have to draw it on one of these boards. So if you guys watched Friday's review, this is the problem is I have to walk away from the computer and now it's not recording my voice. That's why I have a problem with how this setup is, but whatever. Can't do anything about it right now. So if you didn't watch the review over what we learned for the whole week prior, that review has some interesting information that you might want to be privy to. Some people think that the weekly reviews are just me reviewing. That is absolutely not what's happening in the review. If you didn't know, I'm reviewing only the content that people tend to miss on the exams. So that might be useful. And I'm talking about tend to miss, I mean, at least half of the class misses it. So yeah, so you might want to review it. If you're like one of those people who think you don't need it, that's great, good on you. At least go through and make sure you understand the topics that I'm covering. I would say, like, just scroll through and make sure that even on the PowerPoints, the slides, at least make sure you understand those concepts, because there's a reason why I'm telling you about specifically those. So anyways, one of the things that I kind of touched on in the review, I'm going to put up here because I feel like it helps to understand this as we are going through these 2 chapters that we're doing. Actually, the next 3, I'm doing 4, 5, and 7. We're learning about bacteria, then eukaryotes, and then viruses, right? So let's start with microbes in general. All right, so microbes can be divided into 2 groups. Ones that have cells, so we can say cellular, and the ones that do not have cells, right? They're going to be acellular or non-cellular. That is the same word. Don't overthink some of this stuff. All right, so we go over to the non-cellular guys, because they're not doing that right now, so I'll just put it up here for now. There's 2 groups that we have to know that fall into this category. That's going to be the viruses and the prions or prions. I don't care how you say these words, as long as you understand the concept. That goes for all of the words. I don't care how you spell the words in general, unless the question is asking you to spell correctly. But I don't generally care about that stuff. And I'll let you guys know about that at the time. So those are the non-cellular ones. These are the ones we're going to go over in chapter 7. So this is all just chapter 7. You're aware. Okay, so the cellular ones are ones we are starting with today. And obviously, cellular is going to be, what the heck? This computer name. All right, cellular is going to be chapter 4 and chapter 5. And but I'm going to put kind of groups up here. So cellular, these guys can be divided into 2 groups. Start with 2 groups. These are going to be the ones that have a nucleus. I'm going to put this in parentheses because that's not the term, right? This is going to be the eukaryotes. So the eukaryotes have a nucleus. True is what this means, right? And karyo means nucleus. All right, then the other ones are the other group that they do not have a nucleus. Think of the nucleus as like your gateway organelle into membrane-bound organelles, okay? If you don't have a nucleus, you don't have any membrane-contained organelles. That's the rule, okay? So if I'm telling you they don't have a nucleus, I'm telling you bacteria and archaea fall into this category, and that's brought up here. If I ask you guys, you know, where do bacteria make their energy? And one of your choices is mitochondria. That is not the answer. You know that right away because that's a membrane-bound organelle. Now, as far as where they go, you don't have to know that yet, but you would know it was mitochondria. So that's something. So no nucleus. These guys are our eukaryotes. They used to be called prokaryotes. So you can see some of the literature referring to it. Maybe some of my older notes might call them prokaryotes. Pro means before. So it's like the organisms that existed before nucleus did. Okay, but it's the same organisms. I don't know why they changed it. It's been really hard for me to learn eukaryote because I learned it as prokaryote. All right, and then this can be divided into two groups, or eukaryotes can. This is going to be bacteria. And archaea. Okay. And of course, today we're going to be talking about the differences between those two. But we also talked about domains previously in like chapter 1. And chapter 1 domain said we have to know 3 domains, and that is going to be eukaryotes, bacteria, and archaea. Those are 3 domains. So then you understand how they're relating to one another. Now, why do bacteria and archaea, why are they separate from one another in that big of a category? I mean, we're talking about hugely broad categories. In a domain eukaryote, domain of eukaryote puts us, plants, fungi, protozoa, algae, all in the same umbrella, right? That's how broad we're talking here. So then we have bacteria, all the kinds of bacteria under one umbrella, and then archaea. And archaea are so drastically different from bacteria, maybe not with the naked eye, but like looking at them in the microscope, just what they look like. But their chemical makeup, their genetics, they don't have the same kind of ribosomes as they do. Their ribosomes are more like our ribosomes. Their lipids aren't even what we consider proper lipids, like hydrocarbon, like what do you think, like benzene and stuff like that, like octane and stuff. They have stuff like that in their lipids. They're strange, and they live in the most extreme environments on Earth. And we're going to briefly touch on them at the end of the lecture. Like I said, we don't go over them too much. Okay. So that's that. Okay, awesome. I'm glad to hear that. At least you can hear me. I hate that I can't write the notes on here, but okay. So if we're talking about bacteria and archaea and eukaryotes, looking at them like kind of sort of broken down this way. Bacteria and archaea, they don't have the nucleus. Eukaryotes do. That's probably our biggest difference. And if you don't have a nucleus, you aren't going to have membrane-bound organelles at all. We said that. I can click. All right, this is an overview of the structure of a bacterial cell. And for the most part, it's kind of just stuff hanging out in the cytoplasm. If you guys don't know what cytoplasm is, it's kind of the goo that contains the goo in the background of a cell. We right now are people living in air. That's the goo we're living in right now, if you will. And they are, all their parts living in cytoplasm. And cytoplasm is essentially mostly water. It's like 95%, 90 to 95% water. And then there's going to be like electrolytes and just components like sugars and amino acids and things like that that are just hanging out in there. So that's what cytoplasm is. It's a water-based filling. And then we're going to go through each of the major structures and what they do for the bacteria. So bacterial cells, if you're a cell, you have to be able to do those top three things in general. All cells have to be able to do those things. So if we are saying cellular versus acellular, anything that falls in cellular has to be able to reproduce on its own. The single cell has to be able to make another single cell without, cheating, using somebody else's stuff. Metabolism, they have to be able to break down or make molecules for energy, for building new parts, whatever it is, using their own machinery. And they have to be able to process nutrients that are going to give them the energy to do all of this. So, anyways, this is the basics of what a cell would be. So of course, bacterial cells have to fall into that category as well. Some other characteristics about bacteria: they can act as a group. Colonies, we already know that's going to be one cell that just divided a whole bunch into a pile, and it looks like a bump on the media. Then we have biofilms. We're going to kind of get into that a little bit, but that's where they kind of stick up on one another like the plaque on your teeth. Then we have nanowires, which I'm not going to talk about. I don't know enough about it. More or less just creating structures with them and then using those structures for kind of our benefits. But we're not going to get into that. When we're talking about colonies and biofilms, I want to be very clear that doesn't make them multicellular organisms, right? Multicellular organism, you have to have those cells working together to have the functional organism, and that cell has to be with like its organ or whatever it is in order for it to survive as well. So our cells, if I just scrape off your skin cells, are they going to start growing into a whole new person? No. So that's not going to happen. So with bacteria, if you take a piece of a biofilm and then go put it on a petri dish, yeah, it's going to start growing bacteria. Easy. It doesn't care. So they are not reliant. They don't have to be with one another. Therefore, they are not multicellular organisms. Always single cellular organisms. Bacteria and archaea are always single cell. Only eukaryotes can do multi-cell, and only some of them do. Okay, so we have three shapes of bacteria. This is weird how it jumps into this, but whatever. We're looking at the shapes of bacteria, the three major shapes that you guys need to be concerned with. And there's only two I'm ever going to test you on because there's only two that I have in the lab. Two I'll test you on are the top two, but there's another one that's curved. So if you hear me talking later about spirochetes or spirillum, obviously it's talking about spiral shaped. Those are curved shaped cells. But the cocci and the bacilli, already you're probably hearing coccus and saying, oh yeah, I've heard that term, where we're talking about staphylococcus or streptococcus, something like that. That is exactly what we're talking about. In fact, if your organism has that in its name, like we have with Staphylococcus aureus or Streptococcus pyogenes or whatever it is, then you already know the shape of those cells because those are going to be accurate descriptors. Okay, that's why they named them what they are. Not all bacteria, of course, have names based on their shapes and arrangements, like Escherichia. That's not helping anybody out. So, but some of them do. Some of them help you. So if you do see that, if I ask you, hey, lactobacillus lacti, what is the shape of the cell? Well, if it said bacillus in there, then you would know it is rod shaped. So basic concept for these guys, coccus or cocci is multiple. Those guys are round. I'm not going to get super breakdown into little logistics of it. Just think of cocci are round, bacilli are rod shaped. We can also have guys that kind of live in the in-between of kind of ovally shaped, pill shaped, if you will, kind of in between those. And they are called coccobacilli, but I'm not going to put one of those in a microscope and have you try to determine if it's coccobacilli or a bacillus or a coccus, because that's nuts. So don't worry about that. I'll only give you obvious ones when we get into the lab, on the test anyways. All right, so pleomorphism. These are the shapes of the cells themselves, right? You can either be, the cell can be round, or it can be rod shaped, or it can have curve to it. Or you can be pleomorphic. So that's our last option. Pleomorphic means you don't really have a shape. It's a variation in the size and the shape of the cells. It's usually going to be because of structural makeup of those cells. And we'll get into an example of those later on. So here are some pictures of what I'm talking about. You can see in this image that we have in the top, I want to look at the purple, the actual pictures, not the, these are actual pictures. So it's a colored electron microscope image. And They added it on there so you could see like the contrast, but these are round cells, pretty clearly round cells, and they are bunching together in clumps. We're going to talk about what the bunching, the arrangements and stuff are here in just a second. Then we have these round cells down here, streptococcus. You can see streptococcus have round cells, coccus, round, staphylococcus, round. Let's see. They don't have any, yeah, bacillus. Here's streptobacillus, bacillus, rod, and they're just stuck end to end. You can see them there. So you can kind of start seeing the shapes that these organisms can present, and they put them under the category that they would fall into. So the rod base, there's those three in the middle, for example, and there you go. But so those are the basic shapes that we just went over. The cocci and the bacilli are the biggies, the round versus the rod shaped. Okay, so arrangements. If you have your little cocci, little round cells, and your rod shaped bacilli cells, how can they actually naturally be arranged? So first we're going to look at the cocci, and some of these terms are going to cross back over to the bacilli. So bear with me. Diplo. Diplo means two, di, right? So it's just pairs. This is a pretty important one for understanding some diseases. If somebody comes in with symptoms of meningitis and you wanted to determine, you know, what is causing their meningitis, you would obviously take a spinal tap and get their CSF. That'd be the first thing you would do, on top of administering antibiotics. Now, what antibiotics do you give? Because not all bacteria are susceptible to all kinds of antibiotics, right? So do you just shove some at it and hope for the best for now? Yeah, yes, you do. Actually, yes. In this scenario, you do. Because meningitis, sorry. Usually no, you're right. Usually not. But in this scenario, you do. Because meningitis can be so deadly, especially to little kids, that we're just going to lump some antibiotics at you for now and hope that we can keep this at bay until we figure out what this is. Most of the time, if you're going to come in with bacterial meningitis, that's the most serious kind of meningitis you can have, and there's more serious versions of bacterial. You come in there, and those, if you're going to be having severe symptoms, it's probably going to be bacterial. You can get viral meningitis. It's usually pretty benign, and you can just recover on your own. But bacterial meningitis is going to be super severe and uncommon to get fungal meningitis. You can, but there's usually something wrong with your immune system. So like you have HIV or something. So bacteria, if you're having severe symptoms of meningitis and you come into the ER with a high fever and, you know, sensitivity to light, maybe even already having seizures or something like this, they're going to dump antibiotics into you, and they're going to take your CSF and start looking at it because they don't think they have time, right? So that's why they're going to dump those antibiotics in. So there's two major kinds of, there's several bacteria that can cause meningitis, but the two major kinds that you're most likely to encounter are going to be caused by either Neisseria meningitidis, it's pretty much in its name, or something you might not have picked in this lineup would be Streptococcus pneumoniae. Pneumonia is the #1 cause of pneumonia, but it's also the #1 cause of meningitis. So you've heard of the pneumococcus, probably, because they have a vaccine that's available usually to older folks. It's like once you reach a certain age, they want you to get it. They keep contacting me. I guess I've fallen into that range now. And whatever. So turns out that Streptococcus pneumoniae is more tame. It's bad. You don't want it, but it's more tame and more manageable than the Neisseria meningitidis. And that one is called the meningococcus, by the way. So we've got the pneumococcus and the meningococcus. The pneumococcus, the tamer one, the more common one, that one is gram positive. And the Neisseria meningitidis, I can't say it sometimes, meningococcus, that guy is gram negative. And there's one other defining trait. The meningococcus exists in very distinct pairs, diplococci, very distinct. All of the Neisseria species do, whether we're talking about meningititis or we're talking about gonorrhea. So Neisseria gonorrhea can be sort of visualized that way as well. So if I pull out somebody's CSF, the first thing, and you're concerned about meningitis, the first thing you're going to do is a gram stain on the CSF. And if you see gram negative diplococci, it's Neisseria meningitidis. So then you're going to focus your antibiotics on that specifically. And that is the worst one to have. It's incredibly deadly. It's the one that causes the rash. If you've heard about some meningitis causes rash, That's what we're talking about. The rash that's associated with that is not true rash. It's not an allergic response. It is petechiae. It's blood vessels bursting under the skin in reaction to bacteria. So it's quite a severe reaction. You can actually have necrosis develop because of it if it gets too far. And of course, you can die. That's another bad thing that can happen. So knowing things like this can actually help you diagnose, because you don't have to do PCR to get that. Y'all, how long, you're going to learn anyways, how long a gram stain takes. If I were to take a sample of somebody's CSF in here and go do a gram stain, I would have your answer in probably less than 15 minutes. But PCR, you're going to have to wait. It's a minimum of an hour. And that's if your PCR machines are able to accommodate that PCR because it's not like if you have a PCR machine, you can just plug something in it and you get an answer. You have to have primers for the exact disease that you're looking for. Learn about that when you get in genetics, but it's not a save-all thing. Somebody comes in with some obscure disease and you just pop it into a PCR. It's not going to do any good if you don't have the primers, especially if all you're doing is working in a rural clinic and you're keeping PCR machine. You can test for flu, you can test for COVID, you can test for RSV, and maybe some other basic things like meningitis. But what if somebody comes in there and they had, plague symptoms, or you guys might think that we don't have the plague, but we have the plague here in the US. So if they come in with plague symptoms or tuberculosis or something like that you don't usually test for in your rural clinic, you might be able to use some of these techniques to figure out what it is and learn what antibiotic you can give this person, or if they even need an antibiotic at all. All right, because if another thing that you guys are going to learn, if you didn't know this, antibiotics only treat bacteria, no other kind of thing. They don't treat virus. So if you're going to the doctor and they're like, oh, you have the flu, here's some antibiotics, they're either just lazy and don't want you to come back with some secondary infection later on, or they think you have a secondary infection with bacteria and they just don't want to get into the conversation with you. That's literally what it is. They are not treating your flu with antibiotics. That's a lie. So don't fall for that. If you get antibiotics and you have a viral disease, I would personally ask my doctor why they're giving me antibiotics. Because if you're taking antibiotics when you don't need them, you could be contributing to the resistance issue. It's a very serious issue here in the United States, especially right now, because we depend so heavily on antibiotics to keep us healthy, and we misuse them like crazy. So now I'm going to get off track. There's so much we're going to talk about with that on later chapters, so I'm just going to move on. Tetrads, this is just groups of 4. It's literally just groups of 4, like in a square. Staphylococci and I don't care too much about micrococci. I'm not going to ever ask you about that. Staphylo versus strepto, probably a good one to know the difference of, okay? Staphylo is talking about random bunches, like grape-like clusters or irregular clusters. Depends on who you ask. They look basically the same. Strepto is talking about chains, those end-to-end chains. Now, I remember strepto to me sounds like a strip. So I think of a strip of the cells end to end, and that's how I remember that strepto is the chain, and then therefore staphylo would have to be the bunches. So those are the two that most people get confused between, and they're the two that I'm going to test you the most on in the lab for sure. So Sarcina, now tetrads, we had 4 bacteria, little round cells in a square. Sarcina is if you like hook them up together and now you have a cube going on, and they can like be multiples of that as well. Whenever that in real life, in our lab. Arrangements and groupings of the bacilli now. These are the rod-shaped guys. So they can be in diplo as well. So you just have 2 of them stuck together end to end, typically. And then they can be strepto. So imagine if you can be stuck together end to end, you can also just keep the chain going. So that's what strepto will be talking about with the bacilli. Notice we don't have staphylo on here for the bacilli. That's because it gets weird because they're rod shaped is why. So it's like, imagine like rod shaped grapes, that just doesn't stack right. And usually the way that these things are organized are based on how they divide when they create new cells. So that's really what it's usually based on. So you don't see staphylobacilli. Diplo, so they can be in pairs. Strepto, they can be in chains. And then palisades. Palisades is like if you were attached to each other, not by the ends, but next to each other by side. So it's like fence posts. When you look at your samples in the microscope, you can see different versions of these going on. So you might see some diplo, you might see some areas where you will see a little bit of strepto going on, or you might even look in a microscope and you won't see any of this stuff, and that's okay too. Some bacteria don't get in normal arrangements like this, predictable arrangements. It could just say they don't have an arrangement, and that's okay too. I don't really care what the spirochetes do. So then we have the biofilms. Again, I talked about this when we're talking about plaque on your teeth. Another important one medically would be if you are getting like a hip replacement or a knee replacement, they start you on antibiotics before you go into the procedure, right? So you'll be on antibiotics for like probably at least a week, probably before you go in, because they want to be sure that you are nice and saturated with that antibiotic before they're going to cut you open and put this new piece of metal or whatever in your body. That is because they do not want any bacteria from your body, from outside of your body, or from the actual joint itself potentially. It should all be sterile, but we all know that this is wishful thinking. They don't want any of that to start creating biofilms on the surface of the new joint itself when they put it into the body. Because if that happens, it is extremely difficult to treat biofilms in that format, because they're usually, like I said, on the surface of that joint, that metal joint. No matter how many antibiotics you take, you're not going to get them to that metal, right? It's not, you don't have blood flow to the metal itself. So you're hoping that if you give somebody antibiotics, that the other side of it, where your skin is contacting, where we're actually getting infection spreading into the tissue, that just getting it there will be enough. And it can get very difficult because there's usually a lot of different kinds of bacteria that get involved in a biofilm. So we're not just treating one kind of bacteria, and you may not even know what kind of bacteria it is. Even if you culture it, it can be difficult to separate it out. So biofilms can be a serious issue in medicine. We usually are going to most commonly see that in the form of the plaque on your teeth. These guys typically are going to form if there's like a lot of food or safety there or whatever it is, so they can maintain their growth on top of each other. And it really is as simple as what this picture is kind of showing. Hey, we have some bacteria, and you have a nice surface that we can stick to. It's usually going to be porous or have some sort of grab to it. And so the bacteria get their sticky little coating and they stick onto the surface of whatever, your teeth. And then while they're growing, they're being nice and happy and creating more stickiness on themselves, other bacteria can come and stick to them, and then they're all happy in the stickiness together. And they make their own stickiness, this big sticky mess. And that's why it sticks to your teeth the way that it does, because it is sort of just building upon one another of all these different kinds of bacteria, or even fungi can join in, archaea can join in potentially. It just depends. So we can get all sorts of different organisms getting involved in the biofilm once it gets colonized. So the idea is that if you are a bacteria and you're colonizing this area, there's probably good situation going on. You've got food, you've got a good environment, you're safe, you're happy there. However, if you're running out of food, or there's danger or something like that, these guys can send signals to each other to tell each other that the situation's good or bad. If you're having a good old meal, the waste products that you're making from the meal, if you're nice and happy, signal to the other bacteria in the biofilm that everything's going good. However, if you're being damaged, or if you're running out of food, then you might have other things being produced that's not happy waste. And so that signals to the other bacteria in the biofilm that they might want to disassociate from that biofilm and go find some other place to live. So they can actually have that going based on that, which is why it's kind of so important that we floss regularly, because it's easy enough to brush your teeth and keep the biofilm kind of maintained on, basically the visible surface of your teeth. But if you're not flossing, that biofilm will just keep building up on the insides and between your teeth and up into the gum, and can still cause cavities and gingivitis and all sorts of stuff because of the biofilms. So yeah, so floss. All right, so they also can have these appendages, like we have arms and legs, but the things that bacteria can have coming off of them fall into two different categories. The motility one, I say the motility one, because there's only one associated with locomotion. And then the others are whatever else, okay? So it could be usually attachment or providing channels between cells. So we'll go into these. This is the flagella. Well, that flagellum is singular. So flagella are used by bacteria to create motility. You can kind of see in this picture, it's a little hard to see. This is blown up like that, but you can see the little tails coming off of those guys, the long tails. Those are flagella. They cannot control the movement of the flagella in a whip-like motion like you might think. They can only rotate it. They can only rotate it one way, clockwise or counterclockwise. And that's kind of how the little motor-like thing works. And it actually looks like that if you take an electron micrograph of the cell of where the flagella are in the cell, it looks like that. It looks like a motor like that. Really neat. Anyways, it's basically protein based, but it is not covered by the membrane, and they can only control it at the motor. They can't control any other aspect of it. Okay. They just turn it. They either turn it clockwise or counterclockwise, and that's the only way they can move. All right, then we have the ways that these can be arranged on the outside of cells. It could be polar or it could be all over the cell, which should be in a separate little tab, but whatever. So polar arrangements mean that we have the flagella at one end or both ends, but ends is what we're focusing on. And then the non-polar should be peritrichous. Peritrichous is talking about all over the cell. So you can see down here in image B, look at this guy here, man. He's got some wild hair going. So he's got peritrichous. It's all over his surface very clearly. And you can imagine how if you had a whole bunch of them like this, you might actually be able to control your movement a little better. And that's true. They can control their flagella individually. So they don't have to just all at once or none. They can control certain ones, and that can help them, get direction a little bit better than having just one. So that's the peritrichous guys. The ones that are polar, we have the monotrichous. That's the single flagellum, says it mono. Okay, cool. Lophotrichous. This is small bunches or tufts. I don't know what lopho really means, but it sounds to me, it reminds me of wolves. Okay, so when I think lopho, I think of wolves, and now they have nice fluffy tufty fur, and that's the tuft one. And then we have the amphitrichous. It's just both. So you'll have one flagellum at each end. So if you have the tufts, you can have tufts at one end or both ends. It still falls into lophotrichous. But if you have one at each end, amphitrichous. I want to say, I probably won't get into that much detail. I won't break it down that much of a difference, but that is what it means. That's basically how those can be arranged around a cell. Trich is like trick. That's hair, if you didn't know what that meant. So it's like a hair. It looks hair-like, right? Makes sense. Okay. So if you're bacteria and you're moving, how do you decide where you're going to go? They don't have eyes, right? They really don't have ears or anything like that. So how do they know where they need to be going? Well, they move according to chemotaxis or phototaxis. And it's really not as complicated as it sounds, because I'm going to tell you, they move in response to chemical signals. And yeah, it's easy enough to imagine that if you got bacteria living around you and they're making waste, that signals that we're in a happy environment, that we might say, hey, we're still in a happy environment, we're going to stay. That makes sense enough. But even more than that, imagining if I had a plate of freshly baked cookies up here and you guys could smell it and you wanted to come get it. And I said, hey, if you can smell it, come towards the smell and you get a cookie. That is literally chemotaxis. That's chemicals that are hitting your nose and makes you want to move towards it, and you move towards the smell. So that's chemotaxis. And you don't always move towards the thing. Like it says negative chemotaxis, it could be away. If I had a big old plate of dog poop up here, and I'd be like, hey, smell this, and which way do you want to go? And probably nobody would want to go towards it, yeah? So that's negative chemotaxis. They don't want to move towards it. They want it's a repellent, essentially. Some bacteria are photosynthetic, and they would perform their photosynthesis on the surface of their cells because they can't have chloroplasts, right? That's a membrane-bound organelle, so remember that. But they can do photosynthesis. That'll just happen on the surface of their cells. And they, of course, would want to be able to respond to light. So they would have the ability to sense that. But most of them we talk about are going to be the traditional chemotaxis version. Just fancy word for responding to what's going on in your environment. Why is this noise? I keep hearing a boop noise and I don't know what's causing it. Probably whatever it is. All right, so we have how they move. I said that they can only use their flagella in clockwise or counterclockwise motion. So how can they really control their direction? Because they can't control the overall flagellum. You only rotate it or rotate the other way, they go through something called runs and tumbles. If you are running, you're rotating your flagellum in one direction and you're just shooting out straight along the way. A tumble is when you suddenly change direction, and that will cause the bacteria to sort of move around somewhat unpredictably if you have maybe only a few flagella. But if you have a lot, you can kind of get the hang of controlling which way you want to move. So, right. So it says when you're doing your tumbles, you're going to be reversing your direction. This is going to we're going to see a lot of tumbles if you are running away from something. They don't necessarily know which way is the safe way to go. They just know where they are is over here. It can kind of sense that there's dog poop and I don't want to be near it. So if you're going this way, is there still more dog poop in this direction and whatever? So they will do a lot of tumbles to try to survey their environment, so to speak, to get a sense of what the safe direction is to go and let that determine which way they're committing to. So you get a lot of tumbles associated with that. Obviously, if you're going towards something favorable, you're not going to be as concerned with making sure you're going the right way, as long as you're still getting the nice smell, right? So it's not as many tumbles when we go towards something, towards an attractant. So here's the image of that. If we're just chilling and we are not doing anything, they'll just exist and kind of be switching back and forth, whatever. They're never still. If they are modal, they're never still. So they might just be kind of zhuzhing around. I do want to add, because that just reminded me, I do want to say, we talked about flagella being the thing to help bacteria move around. Not all bacteria are modal. So a good number of them are, but some of them are not modal. Staph is not modal. It doesn't have flagella. Okay. Anyways, so yeah, so it'll do these runs and tumbles until it can get a general direction going biased towards whatever or away from whatever. All right, this is our first set of questions. Let's do this. Everybody stand, everybody stand. Let's think about what we talked about here with the bacteria. Which one of the questions was, is this computer janky? It would be yes. Okay, first question. Are bacteria always single cell? And I mean single cell organisms. First victim is going to be Logan. Yeah, all right, good job. So that's going to be yes. They're not multicellular organisms. They can work together, but they're not true multicellular organisms. I just want to be sure I'm not. Yes. So next one. What is the term for a round cell shape? We have Jennifer K. One of these I haven't taken out people who have dropped the class, just FYI, so I kind of apologize for that. Since Jennifer K isn't here, let's do it this way. Jennifer K isn't here. She was part of Charlie. Does anybody from Charlie know the answer to this? Yes, yes. What's your name? Lauren. Okay, so I won't ask you again, just FYI. That's let me write down your name so I don't. Okay, so the next question. I clicked off of it, so of course it's like. Okay, next question we will do, and it's not. Bravo, Jennifer S. What is the term when they are arranged in irregular grape-like clusters? Okay, so we're in the right area. So it'll be staphylo, but at least you're like thinking about like, so staphylo will be irregular grape-like clusters, and the strepto is like the strip, the chain. But we're in the right area. At least we've got those two. Like we're thinking about those. Those are in fact arrangements. So honestly, good job with that at least, because it gets to be a lot. All right, which appendages can bacteria use to move? All right, this is Kaylee. Yeah. All right, good job. And then, what does a peritrichous flagellar arrangement describe? If you are peritrichous, how are your flagella arranged? I keep picking out the same group. So give me a second. Give other groups a chance. This is another Jennifer. Jennifer L. No? Losing all the Jennifers in the class. We have one left. Okay, I think it's Aubrey. Still in this class? Really haven't updated this yet, apparently. I know I've got some new people, so if you're new, I have to make you popsicle sticks. So give me a chance to do that. I'll have them before we get back from Labor Day. Okay, so peritrichus, Rebecca. Becky. Okay. So with this one, we'll say peritrichus, the tri. So here we're not focusing on the tri so much, because that'll be trick that we're looking at here. So that'll be like hair-like. So if we're looking at the peri part of it, and I'll tell you what the options would be: mono, lofo, peri, and amphi. So those are the four possibilities. And so the things you could match with them would be one by itself, one at each end. It could be tufts at each end, or it could be covered all over. So which one do you think? Yeah, exactly. Good job. We got there. All right, last question for this torture session. We have Melina. This is, what is chemotaxis? So that's good. Yeah. So it's going to be movement based on just responding to the chemicals in your environment. It could be the smell for us, right? But yeah, that's it for this session. So you guys can have a seat. All right, let's go down to the boring stuff again. We've got attachment appendages. So we just talked about our motility appendage, and that's the only one that they can use for motility. And not all of them have it. Now these are our attachment guys. If you're trying to stick to a surface, you can use your fimbriae, that's the plural, or you can use your pili or pilus for single. But these guys can provide adhesion. But remember, they are not involved in locomotion or motility. So first, the fimbriae, they look like this is bristles. I think that's a pretty fair assessment. They're like short little hairs sticking off, and they are not as flexible as what we see with the flagella, and not as long, obviously. These are really just protein-based structures that allow the bacteria to stick to something pretty specifically. This is how E. coli will stick to epithelial cells, in particular the ones in your urinary tract, if it's giving you a urinary tract infection. 80% of urinary tract infections are caused by the E. coli from your own bottom. Now you know. Keep it clean down there. We've got pili. These are another one that can be used for attachment. Far more important to me is I would have you guys remember that the fimbriae are definitely for attachment. The pili are the only ones that are going to be involved in this thing that I'm about to get into. So they can form channels between bacteria, so between each other, and then they can shoot DNA between the cells that way. So they can make copies of extra DNA or whatever and just work it through those channels between them. Very effective way of transferring antibiotic resistance. This is how a lot of antibiotic resistance is transferred from cell to cell if it's not just a mutation randomly, which is actually pretty common. We have this long structure made of pilin protein, which I won't necessarily test you about, but it shouldn't be too hard to remember. It's called pili or pilus. And they use it for what they call conjugation. And yes, we talked about, if we're saying conjugation, conjugal visits. So you can imagine it that way. If we're swapping DNA with one another, it is sort of like bacterial sex. So we do call these the sex pili as well. So sex pili are used by bacteria to transfer DNA between them, and that process is called conjugation. All right, let's go into the coatings of these cells. You can have something called an S layer, which is not on your topics of something you know for the test. But it's just a protein coating. There you go. All right, these guys are the glycocalyx and the, which includes the slime layer and the capsule, okay? Slime layer and capsule are a glycocalyx, both of them. And glycocalyx is talking about a sugary, mostly sugary coating, and that is the primary component of it, though it may also include protein. That is very sticky, and that is pretty much the purpose of it for the bacteria. So we were talking about those biofilms forming and that gooey sticky that was on the outside. That's what we're talking about with the slime layers or the capsules. These guys are different from one another, the slime layers and the capsules. The slime layer is pretty loosely associated. You can imagine, like, if I took a bacteria and blew it up inside and put it up here and had a slime layer, it would be all gooing out all over the place. But if I had one that had a capsule that is more dense and closely associated, it might be sticky and a little gooey, but it would be pretty closely associated with the cells. So slime layers are great if you live in certain environments and creating, biofilms and that sort of stuff. But capsules are a little bit more important to us when we are learning about medicine, whether it's human or vet or just, dental, whatever you're going into. So here, let's look at a picture that just kind of shows what I'm talking about. You see how that slime layer is just kind of hanging out off the side, and the capsule is a little bit more tightly bound to it. So you can see in images, and we will be doing a capsule stain in the lab, and it's one of my favorite stains. It's very pretty, and it looks exactly like that. We'll be creating a background with our negative stain, and then our positive stain. We'll stain our bacteria, and then that white in the middle that didn't stain, that's because the capsule doesn't stain. And that will have a halo around the cell. If you don't have a capsule, it would just be the cell in the background. There'd be no white around it. So most of them have a capsule, not all of them. But you can see the difference here. If this was, I don't know what this organism is. Let's say it's staph though. This was staph without the capsule, and then staph with the capsule. You can see it in the actual growth of the organism on that plate. Some of them are pretty clear like that, with that gooiness. So what do the capsules do? If we're talking about the capsules specifically, they, like I said, it's for attachment, which is already good enough for trying to cause an infection in your body, right? So it's already an advantage that we have going on with it. The capsules. But they also allow it to evade phagocytosis. So first of all, yes. Well, first of all, phagocytosis. So this is one of these terms that it's kind of newish now. We'll kind of ease into this term, but I'm going to say it a lot in this course because this is how your body is going to be dealing with invaders like bacteria. If you cut your hand, cut your finger or whatever, and some bacteria happen to get in your body with it. Pretty common stuff, yeah. Even if you wash it, there might be a couple bacteria that get through. There are going to be cells there already ready to go and fight anything that's there. It could literally be like neutrophils is probably the most common ones that are going to come and attack and do most of the work. So don't know what neutrophils are. We'll talk about it in the immune chapter, but they're the number one most common white blood cell in your blood when you get a CBC done. So neutrophils are phagocytes. So phagocyte, phago means eat, and cyte means cell. So if it's phagocytic, it's a cell that goes around eating guys. And we're talking big eating. We're not talking about a couple of nutrients. We're talking about guys that come up and literally just envelop the whole thing and take it into the cell and then digest it into its parts. It's very important for our immune system because then we can take those parts and go show it to the immune system, and then they can attack more specifically. And that's how you get immunity against things. So phagocytes, though, are kind of like your initial soldiers that are going to be going out there getting the evidence of the infection or just destroying it altogether or doing both at the same time. So phagocytes are really important in your immune system. I mean, I can't tell you how important a macrophage is in fighting off an infection. It's probably our most important one. But they're all phagocytes. They all go around and might have little differences between other things they do, but they're all phagocytes. They all come up and eat up bad guys. If we're talking about your B cells and your T cells, the guys that really give you true immunity, they are not phagocytes. They can't. So that's something to think about as well. Cool. So how would the capsule help with evading that? If a big old thing's coming around to eat you up, and it really might be this size difference, or if not more, right? So that cell might be even bigger. If you're a little bacteria and you're swimming around in somebody's body and a phagocyte comes up and wants to eat you up, one of the things that it has to do is like kind of grab onto you in order to get that eating going on. It has to interact with you. That's what I'm saying. If you have enough sticky on your outside, it makes it hard for it to grab on. And this is the truth. So a lot of pathogens have capsules to help evade that so that our cells cannot grab on as easily. Biofilms we kind of already talked about, you can imagine. Sticky helps you stick together. All right, so cell envelope is referring to everything on the outside of the cell. We have a cell envelope Bacteria have a cell envelope. Archaea have cell envelopes. If you're a cell and your stuff's being held inside, then the thing holding it is a cell envelope. So you can have different layers of this cell envelope. With bacteria, you always have a cytoplasmic membrane. The cytoplasmic membrane is the membrane on the very inside. So we have only cytoplasmic membrane. We don't have cell walls, right? So it's just that one membrane holding our stuff in. That's cytoplasmic membrane. Remember, the cytoplasm is the stuff on the inside. So the membrane touching the stuff, most on the inside, the inner membrane, for whatever you are as a cell, it's always going to be the cytoplasmic. So they have to have one of those, all cells do. Then they have a cell wall, almost always, okay? And then they may have an outer membrane surrounding that cell wall. We'll talk about why that is when we get into the differences. But altogether, this can act as the single protective unit, giving structure to the bacteria, allowing them to control what gets in and out, and that sort of stuff. So the main way that we tell the difference between bacteria is through the Gram stain. That's our big step. So it's your first thing you always do anytime you're trying to figure out what kind of bacteria you're dealing with. The Gram stain. You will not stop hearing about it. So, and literally pretty much now that you've learned about Gram positive and Gram negative from these slides, I will always be talking about Gram positive and Gram negative and expect you guys to know instantly pretty much what the difference is when I'm talking about them. Might take a little bit, I understand, but here we go. All right. Gram positive versus Gram negative. When we are Gram positive, that means you stained positively in that. And that means you have a thick cell wall around your inner cytoplasmic membrane. So thick cell wall. And we're going to show a picture in a minute, so don't have to focus too much on the detail right now. If you stained negative in the Gram stain, you had a thin cell wall, and you have to have an outer membrane because that thick cell wall isn't there to help support you. You need another layer to help you out. So think of it that way, okay? So if we're talking about the positive versus the negative, positive is always going to be the thick cell wall. Positive is going to have a thick cell wall. Negative lacks the thick cell wall. So you can even think of it that way. You're testing for the difference in bacteria. Does it have a thick cell wall or not? Well, if it's positive for the thick cell wall, it's Gram positive. Okay, so we can kind of start meshing our thoughts about the bacteria and the results of our Gram stain right away. So let's look at the actual cell walls and see the differences. If we look, we all have this happy cytoplasmic membrane that's on the inside. Great, we all have that. Start there. Look at this. Look at this Gram positive cell wall versus the Gram negative. There's this pink thing down here. That's sad. Okay. So you got this thick cell wall on the Gram positives. I mean, it is nice and thick and just super supportive. It gives great structure as well as protecting against, if I were to imagine that you were like a cell or balloon that is permeable that you put into really salty, even just salt, right? It's going to suck the water right out of it. And that would cause the cell or the balloon to shrivel down, right? It's called plasmolysis when it happens in cells. Well, that cell wall prevents that from happening. It's pretty good at preventing it from happening, right? That is why this should be no surprise to you when I tell you staph, like Staph aureus, or Staph epidermidis, which is just a common one, they're Gram positive. So if I had something Gram negative, they just don't survive as well on the skin because it's salty on your skin. So that makes sense, yeah? So we're already getting to things that are practical about these differences up here. Okay, what are these little bead things sticking up out of the purple guy on the Gram positives? These are lipoteichoic and teichoic acid. I think there's a slide coming up that actually talks about it. But these guys are important for maintaining the cell wall, especially when we're dividing and making new cell wall for another cell. So because there's so much going on with that thickness that they need a little bit of help to manage it, okay? So that's what lipoteichoic and teichoic acid are going to do. When we look at the structure of that cell wall, it's made of peptidoglycan. That peptidoglycan is essentially going to, I know we're running out of time, is essentially going to be scaffolding, and it works like layers that are, like if you think of a layer, and they're all hooked together by the structure of the peptidoglycan. So that's what that it really is, what's going on there, okay? On the Gram negative side, we have a thin layer of that, so it's not as much stability. So we're going to need another membrane on the outside. And that membrane has special lipids in it because it's a whole other new membrane, the gram-negative outer membrane, including lipopolysaccharides, so lipids and sugar units. And that is LPS, if you've ever heard of that, or endotoxin. All of those things are the same. LPS, lipopolysaccharide, and endotoxin. They'll come up quite a lot in the future. All right, so peptidoglycan, most of them are going to have it. This is a depiction of what it looks like. They will just be the sugars linked together, and then across we'll have peptides, so little amino acids linked together to hold them across the way. So that's why it's peptidoglycan. So peptide, and then glyc means sugar. All right, this is just talking about that teichoic and lipoteichoic. If you were to test a patient's sample and it had the presence of teichoic or lipoteichoic in it, then you would already know just from that. I mean, there's other things you could do to figure it out, but even just the presence of that would tell you that it is gram positive. All right, gram negative, we already kind of talked about kind of what it does and why it's there. It just gives a little bit more structure with that, since we lack that thick cell wall. All right, so there's some guys that don't have cell walls and some guys that have weird cell walls. So one of the weird ones is going to be the mycobacterium. Definitely know this one, okay? If you're doing an acid-fast stain, acid-fast positive is mycobacterium, only mycobacterium. All mycobacterium are acid-fast positive, and only mycobacterium are acid-fast positive for our purposes. I know it says nocardia, but I'm never going to ask you about it, okay? So when we say mycobacterium, that actually kind of tells us a little bit about what we're talking about. The mycolic acid on its surface is a waxy substance. It's a fatty acid-based substance, and that is, going to give it protection and hardiness and everything like that. But we can use special stains that are lipid soluble to help us interact with it to see if you have that waxiness on the surface. So this is a special wax. We also have mycoplasmas. Mycoplasmas don't have mycolic acid, even despite the myco, but they don't. Think about plasma. If you took literally a cup of somebody's plasma and dumped it, would it maintain a shape? No, obviously it's just liquid. So these guys don't have a true cell shape because they don't have a cell wall. They lack a cell wall altogether. So they are pleomorphic. The only thing that's stabilizing their membrane are sterols, like our cells have. We're getting there. Almost there, almost there. Cytoplasmic membrane, I don't care about that really. Okay, obviously we're going to talk about permeability more in chapter 9 when we get into osmosis and stuff. But this is typical concept of permeability of what can cross and what can't. Only water molecules and very small uncharged molecules can pass freely across. So that's why membranes are great for keeping everything in and keeping everything else out. This is like LPS. We'll talk about this. All right, this is the gram stain. I do absolutely want to go through this really quick. At least this. Okay, so if you are staining in a gram stain, the first stain you use is crystal violet. Crystal violet sticks in that scaffolding of peptidoglycan. So it'll get caught in there, like sort of like a Plinko machine, or like if you had a whole bunch of webbing and you're dropping balls through it, and a lot of them are going to get stuck, right? You just had one layer, easy that it's going to fall through, easy. You're putting a stain on, and it acts just like a stain, just at first, right? You're getting it soaked into that thick layer on the gram positives. So we're getting in all this scaffolding here. The gram negatives, they'll stain, like I said, but they're not going to hold on to it because it's going to be able to slip out easily when we decolorize. So if you are gram positive and we put iodine on you, that is a mordant, which I always remember like brick mortar that holds things together. The mordant of iodine is going to help stabilize whatever, crystal violet was stuck in there. Now remember, gram negative, it's not actually stuck in there, it's just chilling. It's just sitting there. So we have purple cells at the first step for both of them. Purple cells at the second step, we haven't actually changed the color here. And the third step, we're going to decolorize. This is alcohol. Alcohol is our decolorizer. If you locked in place all that stuff that's stuck in your scaffolding with the iodine, it's not going to rinse out. So it'll stay purple, even with alcohol. However, if you are gram negative, you will run stuff out. Like I said, it wasn't holding onto it for real. It was just sitting on it. So you'll break up the membrane with alcohol. That's kind of what alcohol does. It weakens it, so that helps it rinse out even better. So you are removing the color completely. So you would have clear cells here. That's why we counterstain with safranin, a red dye, so you can tell the difference. That's how this works. And we'll go over again when we learn the gram stain in lab. All right, so whatever we know cytoplasm is to do on the inside, they have circular chromosomes. They're big circular DNA chromosomes. They have one chromosome, one big circle chromosome. You can also have another circle little thing hanging out in you, maybe. These are plasmids. These are extra little bits of circular DNA that you may or may not have that have special traits like antibiotic resistance or ability to make certain toxins. So these are the guys you might make copies of and shoot them across your sex pilus to the next guy. So they're pretty useful in genetic engineering, which we'll learn about in chapter 8. Where they have ribosomes. You need that to make proteins, so of course they have that. Endospores, these are protective structures. They are dormant. I want to just express this very clearly. They are dormant structures that they make in order to survive harsh conditions. So when you're an endospore, you're not actually doing anything. But if you're active and you're happy and you have everything that you need, then you will turn into a vegetative cell. So your normal vegetative cell, hunkering down as an endospore. Think of endo as being inside in the way that like you're hunkering down inside of yourself to hide from something, right? Like duck and cover, or duck and cover from nuclear bomb or something like that. Okay? So, anyways, if you're gonna make a spore, if you are a vegetative cell and you are seeing that, I don't know, Russia's gearing up or something, and you want to make your spores, then you will go through sporulation, and that is going to make that endospore for you. That's the process you'll do. These guys can resist heat, drying, freezing, radiation, and chemicals. They can even resist boiling. This is the problem with things like botulism, which might not be super concerning to you, but how many of you guys know what C. diff is? Ever heard of it? Yeah. If you've worked in a hospital, you know what C. diff is. So C. diff is Clostridium difficile, and it is endospore forming. And that's why it is so hard to control it, because it's pretty resistant to pretty much anything we throw at it. So it can spread through a hospital like wildfire. So that's why you have such strict procedures that you'll have to go through if you do have a patient with it, which is common. So sporangium, if you are if I'm the one that's making an endospore out of myself, then I am a sporangium. Okay, so I'm a vegetative cell, I'm happy. Now I want to turn into an endospore to save myself for later. So I'm going to make the endospore within me. Now I'm a sporangium while I'm doing that. Each cell can only make one endospore. So you like become it, essentially. You transfer your essence into it. So transformation takes 6 to 8 hours to get the endospore made. And then once it's made, it only takes an hour and a half to wake up and turn back into a vegetative cell. Isn't that nuts? So the next time you step on a nail and it's a pretty deep, gouge into your foot and you haven't been keeping up with your tetanus shots, think about this. In about an hour and a half, that bacteria is going to be living and growing in your body, creating tetanus toxin. So stay vaccinated, right? And also same thing with anthrax. Just saying. There's a lot of scary guys that do this, okay? But anyways, if we're exposed back to the thing that we were missing, whether it's your food or water or whatever, then that's what triggers it to come back out. These are some examples of some guys that do this. They're all going to be either Bacillus or Clostridium species. They're the only ones that do it, and all of those do it. So all of the Clostridiums and all of the Bacillus do it. Huh? Lockjaw? So tetanus, it's what they call spastic paralysis. It causes your muscles to like lock in place, and so it locks them down. Typically, if you're going to die from tetanus, which isn't uncommon if you get it, then you're going to die from suffocation because your diaphragm will actually be seized up. Similarly with botulism, but it's going to be flaccid paralysis for that one. They're both Clostridium. Listen to those Clostridium guys. And also C. perfringens is a Clostridium as well. We're not going to do the questions, so just hang in there. Okay. All right, so I don't really care that much about the archaea. They live in extreme environments, okay? And then there's just these terms that I want to go through very quickly. Gracilicutes, firmicutes, tenericutes, and mendozicutes. These guys. You have to know these terms and what kind of envelope they have, okay? So here's how I remember it. This is how I go through it every time, no joke. I will write down all of the words, because I can remember the words at least, but if you can remember the words, you'll have choices on multiple choice tests. So mendozicutes is where I start every time, because when I was little, like I said little, like 11, I had a boyfriend. His name was Joey Mendoza, and that's cute, right? So Mendoza, cute. That's a long time ago though. Okay, I want to be clear about that. I was like 11 years old. We're talking about 30 years ago. So that's a long time ago. So that's going to be archaic. So it's old. So archaea. All right. So that's how I remember Mendoza cutes. All right. Next one that I always go to is firmicutes. That's because firm. So it sort of gives away the answer to me. The ones with the firmest of the outer envelopes is going to be the gram positives. They have that thick, firm cell wall. So those are my giveaways. And then I always have a hard time telling the difference between gracilicutes and tenericutes. The way I've broken it down is if you can get Mendoza cute and firmicute out of the way, you have this one, gracilicute with a G, that I feel like is begging for gram. So gram positive and gram negative. Well, we already put gram positive with firmicute, so that's gram negative. And then tenericute is what's left over. Tenericute, I've looked up, and apparently tener actually is relating to tender or soft. Tenericutes are our ones that lack a cell wall altogether. So they are soft and tender, if that helps you remember it too. Yeah.
So what would a question like identify this type of bacteria?
So the question might be, it could be as simple as, if you had a bacteria that had a thick cell wall, which group would it fall into? And you select it. So that might be your question. That'd be an easy version. But another way that I might word the question could be like, you took a patient sample and you looked at it under the microscope and it was gram positive, and which group would you tell the doctor that it falls into or something? So putting just a little bit more application into it. So that's how I like to approach my questions generally. That's it, I think. Oh, who cares? You look it up to find your organisms. Literally, it's just a book that tells you what your organism is based on features of your organism. I don't care about it, and it makes me frustrated that it even exists and we don't have access to it. But yeah, that's it. So we're gonna go up to, sorry about carrying over, we're gonna go up to the lab now. Everybody just make your way up there and we'll get started right away.